So hi, hello and welcome. I'm Oliver, Microbehunter here, and I would like to welcome you to another Saturday video, Microscopy video. Yes, I put this video, I pre-recorded this video and I put it into Premiere because unfortunately I'm not able to be present live today. But I know that there's so many people joining from all over the world here today, and for this reason I decided I'm going to just stick to the schedule and pre-record something. Of course, you feel free to write comments into the comments section because there is a very lively and active microscopy community out there, and I know that many people join also this Saturday live stream, not only to watch the live stream, but actually also to talk to other people over the chat. So please feel free to do that. Yes, and today I would like to do the following three things. I would like to put three things under the microscope. First of all, a little bit of a follow-up from what we started last week. This here is a water sample where I started to grow some ciliates. So we're going to start with this first. And there are a couple of ciliates in there. I would like to show them to you. The second thing is I received a few days ago a whole bunch of flowers because it's some of my students graduated, and as basically as a little present because I was their teacher, I received some flowers, a whole flower bouquet, and I know that flowers will sooner or later all start to wilt. But I said, well, I'm going to put them under the microscope so those flowers at least are preserved virtually online in YouTube for all eternity. So I'm going to put this flower under the microscope, and specifically I would like to make again a couple of stem cross sections using my microtome. Maybe we're also going to put some other flower parts as well. And then last but not least, look at this here. This is called a catkin, which I kept from springtime. And I would also like to put this under the microscope, specifically this little, so basically this, I'm going to cut off some of this, fuzz over here, and this is also quite nice to look at under the microscope. Okay, so tells us a little bit shows us a little bit on how fine the structures and the details are. Okay, so that's basically what I would like to do today, and we're gonna get started off with this sample over here, and I would like to simply show you what I have done last week. Okay, so if you want to do a so-called an enrichment culture, it's called, I have to focus this a bit. Here we go. Enrichment culture. This basically means you would like to grow a couple of ciliates for microscopy. And the thing is that in a water sample, even though there are many microorganisms present, many of them are in fairly low concentration. So you would like to do the following. You would like to have more of them, so it's easier to observe them. And this is what I have done. You take a wheat grain. I recommend wheat grains and not polished rice because they're simply easier to handle. And again, every time I have to adjust a little bit the focus here, right? And those wheat grains, they will serve as a food source for bacteria, which are also present in the water sample. And then the ciliates, which are much larger, they will feed on the bacteria. And what you should do is you take a wheat grain, and you basically put it on a spoon, and you take a second spoon, and you simply crush it, boom. And that's pretty much it. You see, and those crushed pieces here, you just drop them into the water sample. I have done this last week, and what you will see is the following. As a matter of fact, I dropped in three pieces, and there's one of them over here. There's one of them over here. And this third one is over here, and you're going to see it looks kind of very slimy and fuzzy, and that is indeed the case, because after a couple of days, it's going to look like there's some kind of a slime layer growing around it. So there are basically bacteria growing there, and where there are bacteria, then of course also the largest ciliates, they will feed on the bacteria. So what we have is a small food chain. I simply let it stand there for almost a week, actually, I think around 5 days. And we're now going to put this under the microscope. Okay, and I'm going to show you now two ways of how you can do that. And the first way is you simply take a pipette and you go all the way down to the grain and you suck up a small amount. Okay, and you put that here on the microscope slide. Okay, then you put a cover glass on top, and we'll show you what the problem with that is, because you're going to say, why is this a problem, right? But there is a slightly different way, because what I've done right now is I went all the way to the wheat grain and to collect a sample. So let's have a look at this under the microscope, and let's see what we're able to find here. Yeah, here we go. I'm now using my 10 times objective, and yeah, as we can see, there's plenty of life going on here. Of course, much more than what you would normally be able to see in the water sample. And look at those white dots that you see, those bright white dots. Now, these are starch grains from the wheat, right? And something that you're not able to see quite well are the tiny bacteria that are also floating around in there. As a matter of fact, this sample isn't even that bad because I've already collected samples or pulled samples from the jar where there were so many starch grains that it was almost impossible to actually see anything else. So I'm going to show you now a second possibility, but I think it's fairly good actually. Right, and the second possibility would be that you do not go all the way to the starch grain, but a little bit around it, so that you're actually pulling off more of the slimy material that's around the wheat grain rather. Did I say starch grain? So let's try that. Okay, so I'm gonna use another one over here, another slide, and now I would like to. I have to always refocus a little bit. Maybe not just poke the pipette directly into the starch grain, but maybe a little bit, yeah, more on the periphery. I don't know if this is. Of course, you're probably also going to get fewer cells visible, yeah. But maybe you're able to see them a little bit better because there are fewer starch grains, right? As I just said, we drop a cover glass on top here. Okay, and let's have a look at this. So here we go. Okay, so that is now the one where I did not poke all the way into the wheat grain. Now, the disadvantage, of course, here is that those ciliates are a little bit less inhibited, so they're able to move around more freely because they bump less into those starch grains. So that can be actually a disadvantage as well. Let's go up with the magnification a little bit. Now, the blue background that you see is, of course, because of the specific microscope optics that I have. If you're doing that with a normal microscope, then it would look like this. Okay, it's a little too bright. Yeah. So this is regular bright field. So it might not look quite as colorful, obviously, but you're still able to see everything. Yeah. Just equally well. It also shows that those ciliates that are swimming around are actually not pigmented. If the water sample contains more algae, for example, then they would also look more green because they take on the color of the food that they eat. Yeah. Yeah. So this is lower concentration, obviously. Sometimes you can actually see how they're turning, and then you see that they're actually quite flat, right? So, but let's go on back to the other one. So this one was the one from. Wow, look at this! This is kind of interesting stuff here. I don't know. Yeah. Oh, okay. Yeah. And what you see here is something that I found slightly disappointing, but not surprising. Is that the biodiversity is actually not that high, right? So you see, actually two different types of ciliates here. I mean, of course, I tried to identify them. I was not able to find exactly the same one in my identification book. And the biodiversity is so high anyway that chances are pretty good that they're not in the identification book in any case, in any way. Yeah, but let's move up a little bit. And sometimes it's good if you're actually finding one that does not move quite as much. And then you just zoom in a little bit. Zoom in, well, you go up with the magnification, of course. So again, this is now regular bright field, and sometimes you can also see, some of the vacuoles and other cell organelles that they have. Yeah. So very small in the background, of course, a little bit of biology. Of course, I also would like to explain to you because maybe some of my students are also going to be watching this video. So for this reason, I'm going to turn on, of course, the arrow. And that is I would like to quickly talk about the differences between prokaryotes and eukaryotes, because if you that's the wrong one. I was accidentally moving myself now instead of the arrow. Where's the arrow? Here it is. Here we go. Okay. So what you see over here, those large ciliates, now they're a little bit blurry because they're out of focus. But that's fine. Yeah, so I can put them back into focus, right? But let's put our attention to the small little things that you see here. Those rod shapes here, yeah, those long little line shapes. These are bacteria. These are prokaryotes. So these are of course also cells. They also have DNA. Obviously, all cells have them. Almost all cells have them. Let's be biologically correct here. And they basically don't have a nucleus, and they're much simpler. And those bacteria are growing because they feed on the starch grains here from the wheat grain. that I added. And the ciliates that you see swimming around, they feed again on the bacteria. So what you have is you have a tiny food chain. And this kind of also shows a little bit the huge size difference between those prokaryotes, those bacteria, compared to the eukaryotes, the ciliates that are swimming around here, which have a nucleus. So as a rule of thumb, the volume of one of those ciliates is around 1,000 times larger than those bacteria in the background. Yeah. So, and that is something that you can actually, see quite commonly, but frequently. But sometimes people just ignore those bacteria in the background because, they're simply too small, right? What I'm going to do now is let's go up yet with the magnification, and you're going to see every time when I go up with the magnification, we have a problem, and the problem is the lower depths of field, of course, right? And those cells are simply swimming in and out of the field of view. But here, I want to show you something else a little bit. Again, I'm now completely in bright field, okay? Yeah, so you should be, if you do this same thing, you should also be able to see that. Do you see those lines here in the background here? Yeah, this pattern? Well, these are also bacteria. But these bacteria are sticking to the surface of the slide or the cover glass. Yeah. So yeah, when I made the sample, it was maybe a colony in the water sample, and they kind of were pressed apart, and now they're sticking on the surface. Yeah? So you see that the contrast is kind of low. So what I'm going to do now is I'm going to switch on phase contrast. Yeah. So I'm going to rotate the objectives over, and also have to change something on the condenser here. I open this here, yeah, and this is now phase contrast 40 times. And now you can actually see that phase contrast allows you to see those bacteria much better because they're darker, right? So yeah, not everyone's got a phase contrast microscope, and the disadvantage of phase contrast is it's you're kind of losing a lot of color this way. But sometimes under some circumstances you're able to see things that you're normally not able to see. And look how bright the starch grains now look, right? So that's basically, quite nice. But the problem, let me change the magnification to 60 times again. The problem is they're all swimming a little too fast, according to my taste. Yeah. So let's put on DIC again here. In my taste, they're swimming also. I have to kind of slow them down a little bit. And there is also a technique to do that, and that is you have to add a substance that's viscous, so very thick. And I've got something over here. And this is called methyl cellulose. Here we go, right? I made plenty of videos already about this. It's a powder that you can buy. Methyl cellulose is actually used also in cooking. And you dissolve a tiny amount of this methyl cellulose in water. You have to let it stand overnight because it does take a couple of hours to absorb all of the water. And that's what you get. It's very thick solution, almost like the thickness of honey. You can adjust the viscosity, of course, by changing the amount of water. And the cool thing about this methyl cellulose is that it does not osmotically damage the cells. If you were to add, I don't know, honey, which you shouldn't, then it contains so much sugar that the cells are going to lose water. But this one actually is quite cool because it does not really interfere so much with the cells osmotically, but it really slows them down to the extent it slows them down even so much that they become difficult to see. Okay, so the thing is the following: we are able to see all of those ciliates quite easily because they're swimming around, right? But now, when we actually add this methyl cellulose, we have the problem that they almost will stop swimming, and then we have the problem that they're more difficult to see. So what I'm going to do first is I'm going to take a pipette, and I'm going to simply put a small amount of this on my slide. Sometimes you don't even need a pipette because it's so viscous that actually some of the stuff will actually just stick to the tip. Yeah, look, that's almost like a gel, right? It's probably already more than you need anyway. Okay, and let's now add again a small sample here. I'm gonna just go directly into the specimen again here, just to collect a larger amount. Okay, add a drop here. They're gonna look at this, how white it looks there. So there are gonna be so many starch grains on here, and very important, something that I once overlooked. You have to mix this to really make sure that the methylcellulose is in contact with pretty much all of the ciliates here. Okay, it's still pretty thick. They're probably way too much starch, I think. Okay, who cares? Let's try it anyway. All we care now is simply to see a few of those, and then hope. Look how thick it is. It spreads slowly. And let's have a look now. Okay. Yeah, and this is what we get. Multiple problems. First of all, way too much of the specimen. But look, this might actually be a cell here, right? We don't see any movement, obviously, as expected, because the methylcellulose is doing its job. But the second problem is that there's simply too much material on here, right? Yeah, you see, sometimes there is not. I'm gonna zoom in. I'm just otherwise we're just going to have a close look at this one over here. Here we go. It's too dark. Here we go, right here in the center. Focus a little bit. Yeah. So let's go up with the magnification 60 times. Here we go. Now you can also see the bacteria slowly moving. Okay. Yeah, because the specimen is generally too thick, it's kind of going also up and down, in and out of focus, right? And if you're a little bit lucky, you can actually also see now the cilia, which are the little hair which are beating, right? That is also quite nice. And usually they are beating so quickly that you cannot see it. Yeah, and now you see it's kind of going upwards. Yeah, it's a little bit, yeah. I have to refocus again. Yeah. Then, of course, you can also see those rod-shaped bacteria. Some of them are also moving around, yeah, if you look in the background. So, but I'm not fully happy with this, because indeed there is too much other stuff here, and the layer, the specimen is too thick. So let's try it again with a much lower amount. So again, you see, that's one of the things about microscopy. It's trial and error, right? Sometimes you just have to try it a few times, and then sooner or later you're going to be lucky to actually have a specimen that's quite nice. So let's take again here, over here. Yeah, a little bit less now, and here. I don't know. Maybe there's already enough. Let's try to follow. Let's not go all the way in, but here a little bit on the side. And let's mix it again. You see, maybe it's easier. Yeah, I mean, sometimes you can also pull it up and down a little bit and mix it this way. But maybe not a good idea. Do you see what happens if you do that? If you kind of pull it up and draw it up and down, you get bubbles. Yeah, don't do. So we've got a whole bunch of bubbles as well. You know what? I'm gonna try it in any case. No, let's mix it more. Just want to really make sure. Yeah. Some of the bubbles are going to disappear as we add a cover glass, because they're pushed to the side. So, but that's also something that we've learned now. Don't when mixing it, don't pull it up the pipette. So here we go. Okay, you see that it kind of spreads apart, and maybe some of the bubbles are going to pop. Well, not all of them. We're going to check here on the side. All I need is a cell. I don't need a lot. So let's put this in here, and let's see if we're able to find something here. Of course, every time when you have a new slide, everything is a little bit out of focus again. So I gotta be honest with you, I just checked. I was not very successful in finding a lot of them, but those few that I found, they do move more slowly, as you can see here. So let's go a little bit more. Let's increase the magnification here a little bit more. Yeah, I unfortunately caught not so many of them, but this one actually does now move much more slowly, which is good. Yeah. And it kind of shows that, yeah, as I mentioned before, whole trial and error is really important. But I'm kind of surprised that actually the overall the number of cells is not quite high. So maybe I have missed the wheat grain a little bit. Yeah. So because there are also not so many starch grains in here as well. Oh, there's something over here. What is this here? Here we go. Yeah. Yeah, it's moving not quite as fast as before, but still fairly quickly. I was kind of hoping that it's gonna be kind of frozen a little bit in time, so that you can really see a stationary cell. Unfortunately, is not the case. Another way of limiting the movement, of course, is by simply using very little water. And this, in this case, the cells become sandwiched between the cover glass and the microscope slide. And then this also inhibits the movement. Excuse me, I'm kind of losing my voice a little bit. Okay, let's try a bright field again. You see how much brighter it becomes. The DIC really takes up a lot of light. Yeah. And here we go. You might also close the condenser to increase contrast. Ah, here's another one. Yeah, this one is still moving quite fairly quickly. Yeah, so the idea is you have to play around by adjusting the amount of methyl cellulose that you use, and you have to mix it properly. It could also be that maybe here on the side it was not properly mixed, and that's why they're still moving a little bit faster. But let's have a better overview by really going down with the magnification here. Okay, let's add some DSC for better contrast. You see actually that the number of cells here is not really quite, actually, it's pretty disappointing. Now here's another one. This one actually is now moving slower, so maybe it's in a region where there is higher concentration of methyl cellulose. Over here on the side was a bigger one, and I bumped into the stage, and now I lost it again. Here it is. Yeah. So let's do this. Here we go. Blurry, blurry, better. Yeah, and what I also sometimes, as I said before, recommend is if you give it maybe some algae, some chlorella algae to feed, maybe they're actually going to turn green. Maybe that's also something that I can do in an upcoming live stream. It's just give them something to feed. As a matter of fact, I can actually do this. Today as well. Yeah, and you know what? I'm just going to do that now. Okay, look at what I've got here. And let's try that. I don't know if this actually works well or not, but I'm going to switch back to the desk view. I'm going to start feeding this water sample. Of course, you see it's a little bit greenish, right? Of the algae that were present. But if you want to feed them, what I've got here is a small tablet of chlorella algae. So they're selling this stuff as some kind of a food supplement, but what you can also do is you can take scissors and you can take a small amount and kind of scratch it off in here. Yeah, and then sometimes this also works quite well. So I'm going to then check again after a couple of days. Maybe some of the cells will become a little bit more green. And another thing that I'm going to do now is the following: is I'm going to add a little bit of water because it already started to evaporate. So usually what I do is allow the water to kind of run down on the side of the jar. Okay, simply to replenish the water. And I think that's going to be enough. Okay. Yeah, look, I'm using this black water bottle here on my desk because I don't have, it's a little dusty already, I don't have running water. And the reason why I'm using a black water bottle to store water is to prevent growth of algae. I've also had clear water bottles and then they start to form a biofilm in there. Yeah, so however, there's one concern that I have, and I don't know to what extent it's a valid concern or not. It's soft plastic. I don't know if it's given off some kind of chemicals into the water, which might be harmful, right? So that's something I don't know. Yeah? Sometimes they added some of these chemical substances to make the plastic soft. Yeah? So that's pretty much all. Okay. So let's quickly have a look back here at the as a last little look. Let's remove the arrow. Yeah. So you see now they're actually much better visible. Maybe last high magnification view with my 60 times objective of this cell over here. Where is it? Here it is. It's too dark still. Yeah. And here we go. Yeah, last comment, important. In case you're a little bit, say, well, actually, these are not paramecia, right? It's something else. I want to see those large paramecia. It really depends on what is present in the water sample. I've had, with the same starch, with the same wheat grain method, I've already found completely different organisms. It really depends what's present in the water sample. And over a couple of days, there's a progression. This means one type of species, one type of organism starts to grow and then it disappears. It's succeeded by another one. So there's a progression. And what we see over here, however, in this specific water sample is that at least the biodiversity of those ciliates is not really high. Only found those 2 different types. But there are other water samples where actually I had many more. So it's again, also this is a trial and error. You cannot grow organisms that are not even present in the first place. So a lot of the success depends on the quality of the water sample. And also for paramecia, I have also experienced that they do like algae, right? So very often they can the starch that you add of the wheat grain will increase their growth, but very often they will be next to algae, I suppose, because this is also where oxygen is the highest. Because the algae produce oxygen by photosynthesis. Yeah, I'm gonna call it basically. I call this chapter closed now. Okay, so I've almost half an hour. I want to move now on to the next thing. Okay. So I received a few beautiful flowers. It was during graduation day. Students graduated, and also say thank you to the teachers. As I'm a teacher, I also received one of those beautiful flower bouquets. And the problem with flowers like this is of course, they don't last very long. Obviously. So I said, how can I basically keep them for all eternity? Of course, you put them under the microscope and make a couple of nice videos on them. And then they're at least preserved virtually in YouTube. That's what I'm actually going to do now, is I'm going to put one of those flowers actually under the microscope. Why not all of them? Because not all of them are suitable. I would like to explain this to you first. So this one here is the so-called the Barberton daisy. I need to focus this a little bit. I used an app to identify the flower. It seems to be native in Africa. And it looks like this, and it's very suitable because the stem itself is not lignified. Lignified means it's not woody, so it's fairly soft, so I should be able to cut it reasonably well. And I would like to make a cross-section of this and then put it under the microscope. Okay. There's something interesting that I did notice, and that is if you look at the end here, the stem seems to be a little bit hollow. So what I'm going to do now is I'm just going to cut a piece off here and put it under my stereo microscope to show this to you. Yeah, so I'm using a sharp razor blade now to cut it off. Yeah, here we go. Okay, let's put it under the stereo. You can already see it over here. You see it's kind of hollow. Let's put it under the stereo microscope in any case. Yeah, so here we go. Let's zoom in a little bit. You can see, quite nicely, the stem is indeed quite quite hollow. So this might be a stability problem, right, when I cut it, okay? So I've not tried it before yet. So we're going to simply give it a try, and I'm going to use, of course, my microtome to make a very thin section. At least I would like to try to make a sufficiently thin section. But as you can see, it's fairly soft, yeah? And especially because it is hollow, and therefore I wonder if it's able to resist the cutting sufficiently well. Yeah, okay, so let's cut a part off. I think over here I'm just going to roll it carefully, and it's a fresh razor blade, so it's doing its job quite well. And here, see, on the other side here, I did not cut it very nicely either, so I'm also going to cut this part off as well. If the razor blade is fairly sharp, you almost don't need any pressure. And the other plants in the bouquet were essentially, they had stems that were actually much harder because they were kind of woody, and therefore I think this one is actually much better. Okay. Look at this. I was cutting diagonally. You know what? It actually doesn't really matter. It doesn't really matter because I'm going to put it into the specimen holder here in any case. I 3D printed this here. And you see for different cross sections of stem, I have different. This one is a little bit too big. It should fit fairly tightly, not too tightly. But this one seems to be fine. Okay. Let me focus again here. Okay, so this is the 3D printed specimen holder, and because it's able to slide out fairly easily, what I have therefore done is the following. Over here, there is a needle. Cut the tip off to get it into the right length. Look, there's a little tiny hole here, and I have to push the needle all the way through to hold the stem also in place. Yeah, so let's try that. It's push, 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 push. Okay, and when I do that, then it will actually puncture the stem, and now it's also locked in place. Okay, that's what I want. Okay, so now I'm going to insert the whole thing here into my microtome, and for that I actually set up a separate camera. So here we go. This is it, and I'm going to now put this in here. So what I'm going to do is I have to look down right now, and of course I'm also going to put on my glasses, and let's see how this works. To insert this, and here we go. It goes all the way in. Okay, and notice that I put the specimen right here at the edge, because the edge of the microtome here actually acts as a support as I cut it, right? It pushes against it. So it's pretty far all the way in. I can turn it down even further, so it's all the way in now. And now this is going to be where this is going to be a little bit of a challenge. I'm going to try to cut it like this, okay? And first of all, I have to cut the top off here. Let's give this a try. And you see it's still fairly difficult to cut off, because it's pretty thick. Okay, so I might, yeah, see, okay. Usually the first one is always a little bit more difficult. I don't know if you're able to see it bigger. Yeah, it's unfortunately a little bit dark here from the exposure, but now I'm going to rotate the knob. And I'm going to try to slice off, yeah, until I'm able to slice off. See, it's still too deep. Here we go. Okay, that's the first nice one. And now I'm going to turn it 1, 2, 3, 4, 5, 6. Here we go. Do you see that? Yeah. What I'm going to do now is I'm actually going to put this a little bit into some water, or maybe it's enough if I put it directly on a microscope slide. It's fairly wet anyway. So here we go. Yeah. So 1, 2, 3, 4, 5 is too little. 1, 2, 3, 4, 5. Here we go. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. I think it's yeah. We're gonna see how well this actually works. Okay, let's carefully transfer them now on the slide here. You can see how hollow it is, right? One, two, three, four, five, six. It was a little too thin. Maybe a little too thick. Yeah, as always, you have to give it a little bit of some trial and error, hoping that the more trials you make, that at least some of those cuts are actually going to work. But you see that indeed that the fact that the stem is hollow actually makes it way more difficult. Okay, so I'm going to just put it on here on the slide, and I'm going to now do the following. I'm going to simply put a pretty large cover glass on top, and to check then how well the cross sections actually worked. So I do have also very large cover glasses, but the first impression is that, well, these cross sections are not the thinnest, yeah, but yeah, this doesn't matter. Well, you know what? I'm going to remove the thicker one here, and because I do want the slide to be fairly flat. Yeah, so let's give it a try. Or maybe I can just see it from the side a little bit. It's difficult to see which one is actually thicker. Yeah. Yeah, let's see. And over here I have a fairly large cover glass. Yeah, see a huge one, and I'm going to now add a little bit of simply tap water, and then let's put this on top. So I'm just gonna drop it directly on the cross sections here. Might be a little too much water. That's perfectly fine. I'll simply use some tissue paper to absorb the rest. And a large cover glass goes on top. Here we go. And look. Okay, you see it's okay. I mean, there's quite a bit of air here because those cross sections are fairly thick. But for first observation, I think it's going to be perfectly fine. Let's have a look. I'm already excited. Yeah, I mean, what shall I say? It's a really, really nice cross section. Look at this. Look at those cells here. Yeah, the cross section over here is completely homogeneous here. Here you see even some hair and that's here. That's the hollow space inside. Let's go all the way around it. I'm using now my 10 times, objective. Yeah. So very nice, very nice, very nice. It's a little bit here. It's a little bit destroyed, but that's okay. Overall, I'm very happy. You know what I'm gonna do is I'm going to go down a little bit with the magnification. Maybe we can have a look at a few others as well. Yeah, that's very, very beautiful. We just ignore all of the junk over here. That's yeah. But let's have a look at another one over here. Yeah. Okay. This one it's a little bit ripped apart here, but those here are quite nice. It's over here. And this one here is a little bit thicker. You see that it's a little bit darker already. Here that's the ear bubble at the bottom. Yeah, but it actually shows that this technique works quite well, even though it was actually a fairly soft specimen. But I do want to go up with the magnification now. Yeah, so that is the 10 times. This here is 20 times objective. I'm going to insert the condenser again. Yeah, everything's a little bit brighter. So, and now let's do a little bit 20 times objective. Let's do a little cross section test I'm now using my fine focus knob to see how many cell layers I've got actually. Okay, let's look at the cell walls here. So that is in focus, in focus, in focus. Nice. Look, in focus, still in focus. The cells, they go out of focus and no new cell layer appears. So this means I've actually, yeah, cut a cell layer thick. Let's go continue to the other focus. Yeah, no, I don't see another cell layer appearing. So this actually means yeah, that's pretty nice. Actually means that I've actually cut the cells open. That is also the reason why the cells inside, they look fairly white. Reason being that all of the cell contents has been lost. So now a little bit, sorry, it's got to be a little bit of biology now, okay? So what you see over here is, and I'm going to switch now on don't worry, not too much biology, right? So where is the arrow? So here's the arrow. So what you see, of course, over here, this the outside of the stem. You see it's kind of a little bit greenish still. Okay, that's because the stem is is green, obviously, and contains chloroplasts, and they make the cells green for photosynthesis. And what you see over here, these are the so-called the vascular bundles. They basically contain cells that transport substances up and down the stem. The xylem transports water and minerals up the stem, and phloem transports nutrients, which are sugar, basically, which were made by photosynthesis down to the roots. And usually those larger, thick-walled cells that you see here, that's usually the xylem, which transports water up the plant. And I think we're just going to have a closer look as well. And I can also tell you that, of course, we are looking at a so-called a dicot plant, because of the arrangement of these cells. Dicot, basically, there are two types of the two common types are dicots and monocots. And now we're already pretty high in magnification here, right? Yeah, and yeah, it's a little bit blurry now. It's probably also now has to do a little bit with the thickness of the cross section, right? And those cells over here, usually the thick-walled cells that we have here, these are actually dead cells, long tubes that transport water up to the plant. And if you look now at the arrangement of those vascular bundles, as they're called, you see that they are arranged nicely in a ring. Okay. Yeah. Okay, the center is hollow in this case, but in any case, they're arranged nicely in a ring, and that's very typical for a dicot plant. There are the so-called the monocots, grasses, for example, corn, and so on. They have those vascular bundles distributed all across the stem. Okay. Yeah, so I have to tell you, I'm a little bit happy about that, but I have also something else that I want to try out, and that is I've also frozen those stems, and I would like to make a cut while the stem is frozen. We're gonna see how that works. I've never tried this before, so maybe it's gonna be a complete failure. Maybe not. I have no idea. We're just gonna try it. But before I do that, I have to remove this here. So let's take it out. Okay, you can take it out like this. That's why I made those two holes in here, right? Let's take out the pin, the needle. Here we go. And we can now also remove this here. Yeah, so I just pulled this one out of the freezer. Okay, it is not completely frozen, but significantly more solid. So let's quickly try this one here before it really starts to thaw up, but it is yeah, much more solid. Okay, I have to focus here again. Yeah. So let's give it a try. Yeah, not nice looking anymore. Maybe I'm just going to also start cutting it from the other side a little bit to make it generally easier. And okay, yeah, it's still a little bit crunchy. Okay, and no, let's have it. Yeah, it is harder to cut. It is harder to cut, no question. Okay, next one. Let's see if I'm able to make slightly thinner cuts this way. Up, yeah, I don't know. First impression is might not make a huge difference. But then again, maybe it's not, yeah, completely frozen. It's kind of half frozen. No, this is not something, it's not a new technique. There are indeed so-called cryo-microtomes, essentially where you will cut the specimen while frozen, so that is nothing entirely new. Okay, it's too thick again. No, okay. Okay, I think I'm going to leave it at that. Let's give it a try here as well, just like before. So here we go again. So, of course, freezing is also always a little bit dangerous also because it damages cells. Freezing water, frozen water ice expands and might crack cells open. Maybe less of a problem for plant cells because they have a strong cell wall. I'm not saying it's not a problem. Maybe let's remove the thick one again here. Maybe as in just possibly less of a problem. But you never know. This one also is pretty thick. Let's remove that. Here it's overlapping. Yeah, let's leave it as it is. Okay, again a little bit of water. And of course a cover glass goes on top. Yeah, okay. Another try. What happened now? Okay. Here it is. Let's give it another try. So this one is still the one from before. And where is this? Ah, yeah, that's the one from right now. It's not. This is not a very nice one. This one here is also not so nice looking. Let's have a look here in this one. There are lots of stuff floating around, lots of debris. But look, overall, some of the cross sections do look a little bit thinner. What about this one over here? But honestly, I'm not. Yeah, it is not a huge improvement. Rather, it doesn't look quite as nice. Let's see here. Okay, well, doesn't matter. I'm just gonna go up with the magnification in any case. Let's turn on DIC as well. And this weird arrow, I want to get gone. Yeah. Okay, well, Actually, yeah, zooming in so much is really not a how do you say? It doesn't make it look any better. I think the beauty is are those patterns that you see, but you can only see them, or the arrangement. You can only see them actually um, when you have a lower magnification here. Yeah. Okay, let's go down a little bit. Yeah, I think you get the idea in comparison again to the one that I had before, the freshly cut one. You see, I mean, look at this. It's actually, it looks even thinner, this one over here. Let's go down, yeah. I think I'm just going to stay with the freshly cut one. Yeah. Yeah, looks nice. I'm generally overall satisfied. Yeah, and I think I'm just going to move on a little bit now to the last specimen that I promised you, which is a small catkin, because I just also would like to observe how those how do you call them actually, the tiny little hair, the fuzz on the catkin, how it looks like. Let's have a look. Yeah, so here it is. Again, let's a short look under the stereo microscope. Yeah, here it is. Unfortunately, the contrast is not the very best. But I think we get the idea that we're already at the limit with my stereo microscope here. We're not able to see many more details. I don't expect this to be a very interesting specimen in any way, but I would like to find out in any case. So first, as always, a little bit of water. I need my scissors, and I'm just going to cut off a little bit of the fuzz. I don't want to cut it. Not so easy. Here it is. Here's some fuzz on here. I need something else, another tool to kind of get it off. So carefully scratch it off here, and it goes into the water. Yeah, I know why it doesn't want to go off, because the scissors are not very clean and kind of sticky. So it doesn't matter. Cover glass goes on top as always. Here we go. And it goes under the microscope. Yeah, okay, here it is. This is how it looks like. And yeah, of course, here a lot of air is caught as well. And this is how it looks like. Well, it's still very thin, and up with the magnification here. Yeah, but now we're also able to see that actually there seems to be a little bit of structure present as well. Let's still go up with the magnification. Yeah, almost looks a little bit like it's hollow. Very nice. Look what we're able to find here. Let's. It is actually hollow. I just want to show this to you. You can actually see over here that this is air, filled with air here, and this one now seems to be filled with water, which actually shows that those things actually seem to be almost like a little tube on the inside. Yeah, and here you can also see it a little bit. Yeah. Yeah, just wanted to share that with you as well. Also did not expect that or know that. But that's one of the things that I like about microscopes or microscopy as a hobby is that you always discover a few new things here. Also over here, you see that this seems to be over here water. This seems to be air again. So sometimes the water seems to have entered those tiny microscopic hair somehow. Capillary action, most likely. Huh? I think I'm just going to remove the arrow again. Yeah, that's all I wanted to share today with you. Again, hope you found this video again interesting. Unfortunately, I was not able to respond to your live questions because it was pre-recorded, but I hope that you enjoyed it in any case. I hope to see you again next week. Happy microbe hunting as always, and yeah, enjoy microscopy. Bye-bye.